go. So module one of the 2023 paper. Alright. So the first page, or the first question rather, identify the parts labeled 1 and 2 on amino acid B. What would be 1? Where are my chemists? Where's 1? What's the next part? 2. So you guys can't tell me that that wasn't the easiest set of 2 marks that you've ever gotten in your entire life. Alright, easy. Draw the diagram to show how amino acid A and B form a dipeptide. That not easy too. So this is some chemistry, which isn't hard at all. Right? Easy, easy some. Yeah, and biology is nothing but applied chemistry. So yeah. Alright, so we must be able to do this. One moment, let me just let in this person into the meeting. Alright, so we must be able to draw this, right? So let's go. So we have, let me just, we have a, oh, this is fine. Isn't this methylamine and, well, not methylamine, what am I talking about? This is glycine and alanine, I think. Alright, so let's go. We're gonna end up with, alright, we're gonna end up with something like this. Alright, how do you create that peptide bond? What's a peptide? The peptide is it is a amide, right? So we just need to show an amide linkage. Not true. Not true. I started to get nervous, because I don't know why we're taking so long to respond, but all we need to do is show an amide linkage. This should be a H, not a A. And then we have a C double bond O OH. And then because this is a dehydration synthesis, we're gonna have H2O as the substituent particle there. Alright? What a lovely condensation polymerization reaction we have in here. Right? So lovely. So I have that. This isn't too bad. You guys would be able to draw this, right? You just need to have an amide linkage. Wait, hold on. Nitrogen attached to 3 here. It should be, sorry, 4 here. It should be 3. So that is a linkage. Alright, so that is a linkage that we need to show. Easy piece of something. Let's move on. Alright. Complete the following table by identifying the monomer and the type of linkage for each polymer. By the way, if you're going to the chemistry unit 2, this can appear. It can appear. It's possible. Alright. So it's better we just understand the content from now. Alright. So let's have a look at this. So what's the response that we have here now? So first of all, the monomers that we have here, amino acid is the peptide protein, that's fine. So glucose here, so everything else is glucose. And they never tell you which type of glucose. But the type of glucose for glycogen, that would be what? Alpha or beta? Lovely. For amylose is alpha or beta. And then cellulose would be lovely. Lovely. Right? What type of linkages now? We're gonna have a peptide linkage for cellulose. What type of, go ahead. So we're going to have beta 1 4 glycosidic linkages. Us chemists would just call them esters because they are esters. Right? So it's ester linkages. Right? For amylase no, or amylose rather, this would be alpha 1 to 6 glycosidic linkages, right? Or one. So really and truly, right, it contains both, right? It does contain both. So let me just take this off. It does contain both. So really and truly, it's alpha glycosidic linkages, right? It does contain both. Because no, yeah, it does. Because what we're gonna have here is really and truly branches every for amylose. There are gonna be branches every 12 or so, like around that range, 20 or so linkages, but glycogen is more branched. Alright? Is that the case? Okay. So we're gonna have this would be 1 4, right? G links, and then this now would be 1 with 1 6 G links. Alright? So I have that there. Consider the following procedure, right? That is used to determine the effect of temperature on yeast respiration. Um, I'm pretty sure if I was a biologist, this would have bored me to death. Imagine sitting and watching yeast respire. But that is the case here, right? So, what are we looking for here? We have this entire procedure that we need to study, we need to look at. Alright? Now we're called to, let's see the question now. State one responding and one manipulated variable, right? Now already it's if it's a respiration experiment, we know that the manipulating variable would be the temperature and responding would be the number of gas bubbles seen per minute, right? Before even looking at the steps. So let's have a look at the steps now, right? So we vary the temperature, therefore that is the manipulating variable. Everybody can see that, right? We are varying the temperature. Not true. hmm And then we have the syringe. We're using the syringe. So wait until gas bubbles begin to expel from the tip of the syringe, etc., etc. Right. So we're gonna be looking at the amount of gas bubbles or the amount of gas. We're not looking at the syringe reading, because we're allowing placing temperatures and the entry not to hmm. Okay, we're counting the number of bubbles released per minute. Right? So that would mean now that the manipulated variable, or what we call the independent variable, in this case now, that would be the temperature. So let me just do it in order of the question. The question asks for responding first. Responding would be the number of gas bubbles per minute, I think. Right. And then the manipulated would be the temperature. 2 marks. Right? So just one suitable control for this experiment. A suitable control for this experiment would be what? Go ahead. Well, I wouldn't say so because that would introduce different variables that you would be looking at in it. Because suppose varying sucrose levels would change the rate of respiration. Suppose varying concentrations of yeast vary. Go ahead. hmm So it must have the same amount of sucrose and same amount of yeast. Yes, that is true that you state. Alright? But we're saying that if we have an experiment with different concentration of sucrose, that will be the control. So that's the case, because the control, right? So what is the purpose of a control? Let's ask that first. What does a control do? So I understand what the first person was saying. We manipulate something, something is changed. But would that be the control that we're looking for? Would that be the best control? What is the control in an experiment? Hmm. Room temperature, that wouldn't be the case. So that could be. So we could have one without sucrose, right? But let's say that there's a small, tiny level of sucrose in the water that may affect it. Remember, a control is a group or a sample, right? That is not going to be exposed to the independent variable being tested. It's not exposed to the independent variable. In this case, the independent variable is what? Temperature, not true. And if it is temperature, we could look at something, right, that will cause the yeast to not respond to changes in temperature. So we'll have a sample with yeast, because we're testing yeast. We'll have a sample with yeast. But we need to make that yeast not respond to changing in temperature. We need to make the yeast or something there that is known that will not create any gas bubbles. Right? In this case, right, I think that if you have denatured yeast, right, that would be the best control for this experiment. But there are other controls that could be stated. Alright? Let me just check the chat just in case persons put controls in there that I haven't been reading. Okay. So I think the best control here would be denatured yeast. So what we could do is boil. What I would do is boil the yeast, right? Denatured and keep that as control. So I have denatured yeast as the control, right? Because we would know that that yeast will not respire at all in the same conditions, right, that all other yeast is subjected to. So we're looking at now is the changing of the the rate of bubbles, right, based on the temperature increase. As the temperature increase, the denatured yeast will not respond, but the other yeast would respond, right? So that's what I would put as the control, right? I'm not sure if persons agree though with this. There are other controls that we could agree upon though. Alright? It's getting a bit late. Let's zoom to the next one. Outline your predicted results for the experiment. Your predicted results would be what? Because we're increasing the temperature, So the temperature we're changing from, right? We're going from 20 degrees Celsius to 50 degrees Celsius, well, to 60 rather. Right, so what would be your suggestion? What would be your predicted results? It's like you're writing a PD. What would be the predicted? So we're saying that the variables will increase proportionately, right? Proportionally rather, or no, doesn't matter. Proportionally, right? Until a certain point, right? Or what we call the optimum point. So our optimum point would be achieved, right? And then after that, the rate will decrease, right? Not true. Is not how we normally treat these things, the temperature and a reaction like this. So look at something like that. So that could be the case, or the temperature. That could be the general case there, right? Um. So Describe the induced fit hypothesis. Anyone? What is the induced fit hypothesis? Are we getting tired? Do we need to take a break? Yes. Okay. That's a fair response. I need some persons to add to it now, make it more specific. Hmm. I think there's a fallacy there. You said the substrate will change shape. The substrate doesn't. Substrate can't change shape. So I think you mean that the enzyme changes shape with you. That is what you mean. Okay? But we have to be more specific, It is not the enzyme itself that changes the entire shape, but the active site that changes its shape. So speaking about the enzyme active site changing shape, right? So we're changing the shape of it, right? In what case? To better fit the substrate, right? Upon binding, not true. That's what we're talking about. Binding, that would be the induced fit. So looking at that. But specificity and those things coming with the lock and key hypothesis, right? So would know that. So hopefully we would understand that as well. This is now just looking at organophosphate and the acetylcholinesterase, right? This is a part of the syllabus. It asks us to use acetylcholinesterase as an example, so we must be familiar with this already. All right. So a farmer accidentally ingests organophosphate insecticide. He starts to move involuntarily and experiences paralysis. Because we know that organophosphates are they can affect you neurologically, right? They're kind of like neuro inhibitors. They're inhibitors, right? So the farmer is rushed to the emergency room where doctors administer a high dose of atropine and antidote. Organophosphates inhibit the enzyme acetylcholinesterase, which are neurotransmitters. Alright? If exposure to organophosphates is left untreated for a prolonged period, this can lead to the symptoms such as involuntary movement and paralysis, which were experienced by the farmer. Atropine, right, acts by mimicking the substrate, binding to the enzyme reversibly, right, and preventing the organophosphate from binding. So when we look at something like that, what is atropine? What is atropine? Do you know what type of substrate atropine is? So when atropine binds, right, with the enzyme, it binds irreversibly. So what do we know about atropine there? Non-competitive. The non-competitive inhibitor, lovely, right? So with aid of diagram, explain how acetylcholinesterase is inhibited by organophosphate and atropine. Organophosphates are competitive inhibitors. Atropine is a non-competitive inhibitor, right? Your diagram must be drawn in the space provided on page 9 using the following symbols. You must present your response in complete paragraphs. Lovely. So see it there? Organophosphate is a non-competitive inhibitor because it binds to the allosteric site, right? So seeing something like that. Atropine will act as that as well. So I need to understand that. So we have atropine, right? So binding permanently, right? We have acetylcholine, right? And we also have the organophosphate and inactive acetylcholinesterase. Right? So looking at something like that. So you must be able to draw a diagram that explains how acetylcholinesterase is inhibited by organophosphates and atropine. Wait, so based on the information here now, it shows that atropine is non-competitive, but in the diagram here it's showing that it is competitive. This is weird. It says that when atropine acts as a substrate binding to the enzyme reversibly, my apology, it should be irreversibly. I thought this said irreversibly. So since it is reversibly, atropine is the competitive inhibitor. So that is the competitive inhibitor, while the organophosphate is the non-competitive inhibitor. So all you have to do is use diagrams to explain that. That's all you need to do. Atropine will bind to the active site here. Organophosphate bind to the allosteric site here. I believe that you guys have enough information to respond to this question. Alright? Make sense? Competitive binds to the active site. Non-competitive binds to the allosteric site. That's the difference. Right? So since the atropine is shaped like this, using the lock and key hypothesis, we'd know that the atropine would be a competitive inhibitor. Right? So we know about that specifically, while the organophosphate would be non-competitive inhibitors. Alright? So all you need to do is explain how that works, how it is inhibited. So competitive inhibitors, right, would inhibit by binding to the active sites, right, and preventing other substrate from binding to active sites, right. While the non-competitive inhibitor would bind to an allosteric site that will now augment or change the structure of the active site on said enzyme. Alright? So we must know the difference in their function, what they do. We must know what they do. Competitive versus non-competitive inhibitors. That is what the question is about. The question is not about anything else. Right? So let's provide a diagram and explain the difference between the competitive inhibition and non-competitive inhibition. Right? And that's 12 marks. That is 12 marks.